I took the liberty of just tweaking the title of my talk a little bit, and I'm sort of going to mix it up. And actually, after speaking to some of you out there today, I actually, during one of the sessions, added a couple extra slides and some things to even highlight some things that I wanted to add in. So I'm going to speak to you about really two aspects. One is the genomics of KIT and PDGFR alpha wild type GIST. You've heard a lot about KIT. You've heard some about PDGFR alpha, but not a huge amount about some of the other subtypes of GIST that some of you might have or be aware of. And then I'm going to talk to you about the succinate dehydrogenase or sdh-deficient GISTs. So we've really seen this evolution over the years of the genomics of GIST. You know, back in the early 2000s, we were talking just about KIT. Oops. And what's happened evolved over time is that we've suddenly started realizing with increasing genetic testing of tumors, as well as just increasing access to our ability to do this in the clinical setting, is this evolution of really starting to think about the different subsets of GIST. So if we look at the timeline of what's evolved, as you heard earlier today, so the discovery of the gain of function mutations in KIT occurred in 1998, and then Dr. Heinrich in 2003 discovered and described the PDGFR alpha mutant GISTs. And the way, as you've heard, these sort of are these receptor tyrosine kinases, and they sort of signal down these pathways where basically when one of these receptors comes together, they signal downstream. And so that's sort of, you'll see is a little bit of a theme that's recurrently comes up as we've sort of discovered more and more genetic alterations that are driving these tumors. So in 2006 was the first description of germline neurofibromatosis 1 gene mutations, or NF1. So this was germline being those patients that have a hereditary version or basically inherited from parent to child. These are often multifocal GISTs that most often occur in the small intestine. I mean, besides one case I've personally seen occurring in the stomach, every other single one I've ever seen was in the small bowel. These only represent a small fraction of GISTs associated with NF1. While the biology can be heterogeneous, most of these tend to have a very sort of indolent or slow-growing biology. More than likely, they may not respond to imatinib, but potentially can. And that said, we don't have any drugs that actually directly target NF1 at this point. The other thing, and that sort of I added in here after talking to some of you out there, was there's actually a secondary thing we discovered several years ago, that sometimes we can actually see these NF1 mutated GISTs in the small intestine that are not inherited but somatic. Somatic meaning that it occurs just in the tumor, but it wasn't inherited from one's parents. And this was the first case was described in 2015 by the group at Fox Chase. And we went on to then describe in further detail about the biology of these and really trying to understand and figure out sort of how do we predict which of these patients is going to have an NF1 mutant GIST. And what we discovered is that about 5% of GISTs occur at this area, which is basically we call the duodenal jejunal flexure, which is basically this transition from, as you've sort of heard, esophagus, stomach. Duodenum is the first portion of the small intestine, and then the jejunum is the next portion. So right at the transition from the duodenum to the jejunum, what we started noticing as we were taking care of patients was that we were frequently seeing these mutations in the NF1 gene, often in the absence of having a hereditary NF1 syndrome or neurofibromatosis. Occasionally, we were seeing actually both as well. And we were actually even seeing some of these patients that had KIT mutations. And so this diagram basically is sort of a co-mutation plot where basically each column is an individual patient. Each row is either some type of biologic factor of the tumor, like the mitotic index here, as you heard about from Dr. Lazar, or the genes mutated. And what you can see here is actually, when we looked at our data and combined this with cases from Memorial Sloan Kettering, we actually had a large number of patients that had not hereditary, but somatic, meaning non-inherited, mutations in this NF1 gene. And some of these were actually associated with mutations in KIT, as you can see here, or even BRAF. And I'll get to BRAF here shortly. So what we learned from this study was that this sort of duodenal flexure, or surgeons will often call these the ligament of Treitz GIST, frequently occur in this region and have mutations in this NF1 gene, whether they're somatic or germline. One of the other things that we learned is actually, we were sort of evaluating this, we recognized that some of these patients actually didn't have all the sort of syndromic features of germline NF1 or inherited NF1, but actually had a mutation in their NF1 gene that they had inherited. And so this was really the first kind of evidence that they had a finding consistent with the hereditary syndrome of NF1. So sort of piquing our thought that we should be thinking about this location as sort of a biomarker for these NF1 mutations, but also potentially occult or sort of unrecognized germline neurofibromatosis needs to get worked up if a patient has a mutation. And then finally, as I noted before, we discovered that several of these patients actually have mutations in KIT and the NF1, which would then potentially portend that they may respond to drugs that are targeting KIT or BRAF. The next sort of evolution, and I'm going to talk about this more later, was the germline succinate dehydrogenase, or SDH genes. And I'm going to sort of skip ahead of that, and then I'll circle back. BRAF was the next discovery in 2008. And so as I mentioned, you can sort of start seeing this theme. So KIT and PDGF signal through this sort of RAS pathway, which is actually blocked by NF1. And then the next step in this downstream is BRAF. So not surprisingly, sort of as we sort of follow down this downstream pathway, we start seeing mutations in the same genes that are causing GIST. So these BRAF mutant GIST tend to be in the small bowel. They tend to express KIT, just like we've seen, and they tend to have sort of variable biology. Some can be sort of more slow-growing and indolent, and some can be more aggressive. This was actually the first patient that was actually treated here at MD Anderson, published in 2013, that had a BRAF mutant GIST that was targeted with a drug that actually targeted BRAF. At the time, it was just a bunch of numbers and letters before they actually came up with the name dabrafenib, as it was ultimately FDA approved. And what you can see here is there was a modest shrinkage of the size of this tumor. But what's evolved over time is that now there's actually an FDA-approved combination of two drugs for BRAF V600E mutant tumors in general. So this is not just GIST, but any tumor that has a mutation in this gene, which most commonly we actually see in melanoma, but you can see it in other tumor types, including GIST, is a combination of actually two drugs. One is dabrafenib, which basically targets the BRAF right here. And the second one is a drug that targets the MEK, which is immediately downstream. So you're basically blocking the downstream signaling at 2 points with this combination. And so that's the FDA-approved combination now for this type of GIST. Now, 6 years later, we get into the SDH epimutant ones, which I'll get into a little bit later again. And then in 2016, our group, working with Dr. Heinrich, described what was at the time thought to be quadruple wild type, meaning that they were negative for KIT, they were negative for PDGFR alpha, they were negative for the sort of RAS pathway being these BRAF and NF1 mutant GIST, and they were negative for SDH. And what we described was that there was fusions of two different genes actually coming together sort of and melding into one that was actually driving this NTRK gene. And not surprisingly, these NTRK genes sort of look a lot like KIT and PDGFR alpha. There's sort of these receptors that basically signaled through very similar pathways downstream to regulate the cells. And so this was actually a patient, one of the patients that was in our original study that was actually published here at, treated here at MD Anderson, had actually failed 5 lines of therapy. You can see this PET scan is pretty dramatic with the amount of disease here. That's all lighting up. Large tumor here in the liver. This patient had failed 5 lines of therapy at the time, went on a drug to target the NTRK fusion, and you can see here 6 weeks later had a dramatic response. Now, what we now know is about 5% of the NTRK fusion tumor types are represented by GIST. And we've also seen that ultimately from the clinical trials, we've actually seen several responders with including actually complete responses, meaning 100% resolution of tumors of treating patients with NTRK fusion GIST. Now that said, there's actually some people that have actually raised the question, does it really exist? And sort of questioning whether these were truly GIST or not. This paper actually just came out this week in Clinical Cancer Research from our group, sort of trying to put this answer to rest. This was a patient that we had treated in San Diego. Actually, a patient was treated across town, had several operations, several times of treatment of imatinib, kept recurring, came over to see us, had a clear diagnosis of GIST with a KIT staining, DOG1 staining. But it turned out when we did her mutational testing, she did not have a KIT mutation. She had an NTRK fusion. We put her on that drug, larotrectinib, and what you can see here is this sort of nice response both around her stomach as well as down in the pelvis. These are sort of this plot basically shows sort of the shrinkage of the individual tumors. She had 5 different tumors, and you can sort of see that some of them only shrunk a small amount, some of them shrunk a lot. But ultimately, when I took all of these tumors out, what you can see here is actually they were 99% dead, only 1% viable cells. So sort of putting to rest this concept that there are GISTs, and when we go back through the literature, there are, including some of the cases we've published prior to this, but now including this new case, there are clearly defined evidence that these do exist. There's been some question whether some of these other ones may or may not have been, but in the end, I think the answer is yes, they are there, albeit rare. The final sort of subtype I'm going to talk about are the FGFR fusion GIST. These are the fibroblast growth factor receptor ones. These were the same publication we published describing these gene fusions in GIST. And this FGF receptor is very similar again. You know, sort of you're noticing a theme here of a lot of these sort of pathways, all sort of signaling down from a receptor down through these similar signaling pathways. And so we actually now have drugs that target these FGFR fusion GISTs as well. These are pretty rare. We don't see them very often. But what we're starting to see is now this sort of fractionation from where I started out and said, like, it's all KIT, to now we're starting to sort of subdivide, and I'll talk more about the SDH ones coming up next for the rest of the talk. We've talked about the BRAF, we've talked about the NF1, and then that sort of quadruple wild type group are starting to break down into all these little sort of subsets of GIST with different alterations that are driving the biology. There are still a subset that we don't understand. You know, I wrote this paper several years ago as sort of a call to abandon the term wild type, because ultimately we're going to figure out what's causing each one of these as we do more analyses, more understanding of the molecular biology, to really try to ultimately define what are these sort of unclassified ones. It's still a small fraction of GIST that we can just say, hey, we don't know, but time will tell us. But the biology of GIST extends beyond just the KIT mutations. What we actually are starting to understand now as we sort of think about this further, and I think that example of these tumors that I described to you before, the NF1 mutant GIST right at this duodenal jejunal flexure, started sort of bringing up another concept in my mind, was that there are certain areas of the GI tract that are sort of have a predilection for certain mutations, suggesting that we still don't fully understand the biology of this disease. And what I mean by that is while we see the KIT mutant GIST occurring everywhere from the stomach all to the small intestine to the colon and rectum, certain ones like the PDGFR alpha mutant GIST really only occur in the stomach. The SDH ones that I'll talk about really mostly only occur in the stomach. The gene fusion ones I just described, these NTRK fusions and FGFR ones, are mostly in the small intestine. The germline NF1, small intestine again, the BRAF and this sort of somatic NF1s are most commonly right at that one spot, that duodenal jejunal flexure. Now, we can even take this even a step further. As what we start realizing is actually within the stomach, that there's even portions of the stomach that have a higher predilection for one mutation type versus another. And what became obvious to us is that when we start looking at the stomach, the top part of the stomach is almost 99% KIT mutant. So if you've got a GIST arising sort of in this area, and the sort of challenging area that Dr. Ascali was just talking about, the esophageal gastric junction, these are almost all KIT mutant, suggesting that most of the time, probably a lot of those will respond to imatinib therapy. But as we go more distally in the stomach, sort of farther down, you can see here along the lesser curve and along the greater curve, you can see about 25% of these GISTs in both groups are non-KIT. So they're, whether they be PDGFR alpha or the SDH ones. Interestingly, when you get to the bottom part of the stomach, we call the antrum, or the sort of pre-pyloric region, that sphincter that he talked about before, actually only 25% of them are KIT mutant. Now we're talking 75% are these other mutations. And so it can sort of raises lots of questions about the biology of these, the predilection for certain locations, even potentially the cells of origin that might be perhaps a little bit different between these cells. So now I'm going to sort of focus on the sdh-deficient GIST, and for the majority of the rest of the talk is really going to focus on these, because that's where in the last several years, we've made a lot of progress in the field. So by way of a little bit of background, so the SDH1s are described as SDH deficient because, as Dr. Lazar explained to you earlier, they lack expression of that SDHB staining. And the SDH genes, there's basically 4 genes. There's SDHA, SDHB, SDHC, and SDHD. And basically during metabolism of sugar, they convert a molecule called succinate into fumarate. And this occurs sort of in the powerhouse portion of our cells called the mitochondria. So these are not oncogenic mutations like you heard about from Dr. Samaya at the start of pushing the gas pedal. The biology of this is very different from that, okay? These tumors are also unique in that they represent only about 10% of all the GISTs, and very different than most of the other GISTs that we've talked about today. Besides spreading by blood or sort of by peritoneal surfaces, meaning inside the abdominal cavity, they have a propensity to spread through the lymphatics to lymph nodes, and that's typically not what we see with every other GIST we talk about, with the exception of some of those fusion GISTs. The NTRK fusion GISTs sometimes will go to lymph nodes. So these SDH-deficient GISTs really come in 2 sort of subtypes, if you will. There's the so-called Carney-Stratakis syndrome, or some others will call it the hereditary paraganglioma pheochromocytoma GIST syndrome. These are caused by hereditary, meaning inherited SDH mutations that an individual receives from their parents. These are often occur in adolescents and young adults, to what we thought for many years. They're associated with paragangliomas, which is another type of tumor that patients can get, which are basically hormone-secreting tumors that can occur anywhere from the neck, the chest, or inside the abdominal cavity. And they make hormones like adrenaline and sort of derivatives of adrenaline. Now, the latest thing, the sort of secondary group that was discovered in 2014 and described by the group at the NIH was a so-called the Carney triad, or SDH, now we call SDH epimutant tumors. And what's different about these is it was becoming increasingly recognized for many years that there was young girls, sometimes adolescents, even younger, generally not males. To my knowledge, I think I'm only aware of 1 male that's ever been described to have had this, so pretty much almost non-existent. That basically did not have a mutation in these SDH genes. And what the group from the NIH described was that these patients have what's called promoter hypermethylation. So when you look at a DNA, there are these little sort of methylation black marks here. And when those black marks become increasing in number, it basically prevents the DNA from being converted into RNA and ultimately into protein. So it's basically turning that gene off, if you will, the more black marks you have. And so what was described is that these patients with these SDH hypermethylation was what we call it, basically are turning off the SDHC gene. So anytime one of these genes is either messed up by a mutation or by just turning off one of these genes, the whole complex falls apart. You need every piece of this puzzle, all the A, the B, the C, the D, all has to come together and be present for this process to work out and for them to be together. If one is altered in one way or another, either being silenced or being mutated, this doesn't work out. Therefore, this complex falls apart. Therefore, when the pathologist stains for the SDHB staining, it doesn't exist. Therefore, we call it an SDH-deficient tumor. So I said to you that, we sort of have, our understanding of this has really been from small case series and really trying to understand the biology of this. And there's been many sort of descriptions of this as often being these SDH-deficient GIST being indolent, pediatric, insensitive to the tyrosine kinase inhibitors that we typically think of, recalcitrant to serial or more extensive surgical operations. And yet at the time, as we were sort of seeing this more and more in my practice, I was seeing more and more patients dying from this disease and asking, is there really a more aggressive biology than we're appreciating? And this led us to actually form a consortium with the NCI and with the liferaft group to start thinking about how do we pool all of our data together to really start looking at these patients and understanding the natural history. And so the first thing we did was actually we pulled together 256 patients from around the world with SDH-deficient GIST. And we're actually right now, I'll tell you, we're in the process of merging our data set with another data set from a group in Europe that has another 140. So we'll be up somewhere close to 400 patients with SDH-deficient GIST, which will truly be the sort of the largest analysis that will give us really the sort of most definitive perspective we can have of the biology of this and the natural history of this disease. To my surprise, actually the median, meaning like the middle point sort of age for these patients was actually not teenagers. It was actually 29 years old. But in the series, we actually saw patients all the way from first diagnosis at 5 years old to last diagnosis at 79 years old. If you would have asked me this a couple years ago, I would have said, that's probably not possible. But the reality is, as we've sort of looked more and more, we're seeing it. Probably, so we always thought that if you sort of escaped maybe your, into your 40s or so, you probably weren't going to get it. It's not actually true probably, but just less likely. The other interesting fact is unlike you sort of heard earlier that there's generally a sort of equal male-female predilection for GIST in general, you can see it's about a 2/3, 1/3 split for SDH-deficient GIST. Now, the natural history of GIST, and this is looking at the overall survival. This is one of those survival curves that you've seen many times before. Sort of just as a reminder, sort of the flatter it is, the better it is. Every time this little steps down, that means somebody unfortunately passed away from their disease. If you look at the median, meaning like the middle point, so when 50% of people have passed away and 50% of people are still alive, was actually turned out to be the median of 39 years. So at a sort of 30,000-foot view, you'd say, yes, this is an indolent biology. But if you look a little bit deeper, what you start seeing here is that if you look down here, this is how many people have passed away at 5 years. This is how many at 10 years. So at 5 years, 11% of people have passed away. At 10 years, 77 or 23% have passed away. So, and then you can sort of see things sort of flattening out with time. But raising the question of, 1 in every 4 of these SDH-deficient GIST patients may not be surviving all the way out to 10 years. And so maybe there is really 2 flavors of the biology. There's the indolent ones that people live for a long, long time, and there may be a more aggressive biology. And how do we start understanding this was the next question. Now, in 2020, we had, several FDA-approved drugs in our armamentarium for GIST. Imatinib, sunitinib, regorafenib, ripretinib. You've heard about all of these. Now, if you look at this if you look at the patients where I specifically went to all the different trials and studies that have been published and pulled out all the SDH patients that were known, you can see here that the response rates, the objective response rates, whether it be complete responses or partial responses, meaning a greater than 30% shrinkage of tumors or 100%, like complete lack of tumor there, the response rates were not great, okay? And the largest study was like 3 out of 16 patients, and regorafenib had responded, really. Ripretinib, there was very limited data. There was only 2 patients. Now, around that time, there was also had been 3 different studies that had been performed specifically on sdh-deficient GIST patients. And these studies were done to target different molecules that had been discovered in the laboratory to been overexpressed or basically be potential targets. And this included drugs lincitinib, vendetinib, and guadocitabine. And unfortunately, when you look at the data from all these trials, there was not a single responder there. So, this is only 5 years ago, I'll remind you. So one of the things that we set out to do in my laboratory, and as being a surgeon, I'm operating on these patients. We can get tissue from these patients. We took it back to my laboratory. After a lot of trial and error, we were able to figure out a way to actually start growing some of these sdh-deficient GIST in the laboratory. And up till that point, there were no models that we could really use that could replicate the disease exactly. There were ways that we could sort of get rid of this gene in an individual cell line, but not specifically in GIST cells. And so with these sort of new armamentarium of these patient-derived models that we could start studying, we started looking, and we basically realized that, number one, these tumor cells could sort of recapitulate the biology of the tumors, both metabolically as well as at the RNA expression level. We were able to start growing these cells from an SDHA, an SDHB, and an SDHC patients. We could grow them in 2 dimensions, we could grow them in 3 dimensions, and then we could start treating them with drugs. And this is just a reminder up here at the top, the response rates for imatinib and sunitinib in these SDH patients. And what you can see here is the way these are sort of cell viability killing curves. The higher doses are to the right, and this is the cell viability. The quicker this drops off, the more sensitive the tumor cells are in the Petri dish to killing. So you can see here, they're not very sensitive to imatinib, a little bit more sensitive to sunitinib. Now, one of the things at the time that we recognized is that elevated levels of that molecule succinate can actually affect DNA damage repair. So anytime our DNA is damaged in our cells, our body's trying to fix it. But if that fixing doesn't occur, then it actually can sometimes lead to cells dying off. And so there's drugs that we can actually use to target that. Well, it turns out there was a drug that's called temozolomide. It's a DNA damaging agent. Turns out actually in the early 2000s, there was two trials, each of 18 patients that used to treat GIST in a clinical trial. One was done here by Jonathan Trent, who unfortunately couldn't be here today, and the second one was done in Europe. But what you can see there is in patients with GIST treated with temozolomide, there was not a single responder. But around the same time as we were sort of working on this in the laboratory, there was a study that came out identifying that patients with SDHB mutant paraganglioma, that relative of GIST, in a small cohort of patients, 50% of those patients actually had a response to temozolomide. In the meantime, we were actually trying to look for discovery of drugs that could target in our cell models. And lo and behold, you can see here, as we start increasing the dose of temozolomide in our cell lines, we start seeing increasing killing. Using a different model that actually Dr. Heinrich had developed in his laboratory, where he could actually sort of get rid of the whole SDH complex in cells by just knocking it out. What we could show is actually when we treated sort of normal cells with temozolomide, they didn't die, or you can sort of see in these flat lines here. If we got rid of the SDHA gene, they became sensitive to the drug, as you can sort of see here also in the red. If we rescued them by giving them back the wild-type SDH gene, they became resistant again to treatment, as you can sort of see here on this graph as well, suggesting that perhaps the loss of SDH may be a biomarker for predicting response to temozolomide. And so around this time, actually, Paul Fanta, who's one of my medical oncology partners at UC San Diego, we had a patient who had failed all the other FDA-approved lines of therapy. Ripretinib was not available yet, We didn't really have many other options left, and we put this patient through a clinical trial protocol on temozolomide. And what you can see here is he had a nice response to treatment. He actually had multiple small tumors here that you can't see any longer. And this larger one, you can see shrunk down pretty significantly. And so what this led to actually was a phase 2 clinical trial of temozolomide in sdh-deficient GIST led by one of my other partners, Adam Burgoyne. We opened this up at 4 sites at the time, including Dr. Heinrich had it open up at OHSU. And what we saw here was that we actually had a 22% response rate. We actually treated 23 patients. As you can see, compared to the sort of other sdh-deficient GIST trials, this was sort of the first signal that we had something positive going on. What you can see here, actually, when you sort of look in more detail, is we actually had some nice responses in a couple of the patients. You can see, interestingly, there are 2 of our 3 best responders were actually in green here, were sdh-B mutant GIST. We had several patients. We actually had 5 patients that had greater than 30% shrinkage. And we actually had another 3 patients which had greater than 20% shrinkage. And albeit it doesn't technically hit our threshold of 30%, which is somewhat of an arbitrary number per se, I'll take 20% shrinkage over 0% shrinkage or growth any day of the week. And so it's suggesting that we've sort of, have a potential for a new therapy to think about for patients with sdh-deficient GIST. And so at the time, this was the largest clinical trial conducted in an sdh-deficient GIST patient population. And what we found was actually the temozolomide was actually well tolerated. And it actually we sort of had an efficacy signal that was for the first time. As I'll talk about in a few minutes, it actually opened up the opportunity for surgical involvement at that point. I'll talk about that more. But given the lack of other effective treatments at the time over the last couple of years, this sort of potentially meets one unmet need in this patient population. But unfortunately, I'm also I'm glass half empty kind of guy, and so I'm saying to myself, well, great, it worked in roughly a quarter of patients, but what about the other 75%? How do we do better? And so we went back to the laboratory, and one of the things that we did was we did these sort of protein analyses where we discovered that there was a protein called DR5, or the other name for it is death receptor 5. And when this death receptor gets turned on, it causes cells to die. And it turned out, actually, when we treated our cells with temozolomide, it caused upregulation of this protein. And so we went back and looked in our cell lines, and lo and behold, it was there. We looked at using immunocytochemistry, basically looking at the cell lines, and red staining here tells us that this was not expressed in the control cells, and when we treat it with temozolomide, it gets turned on. And actually, this is actually from patients on the temozolomide trial. We can see it's actually the protein is getting expressed here in brown. So it turns out there's actually 2 places in the world that have drugs that target DR5. One is in the Netherlands, and one, it turns out, is a spinoff from UC San Diego. That's 3 miles from my laboratory. And so this company is called Inhibrx. I had the pleasure of having dinner with one of the co-founders one night, and within 2 weeks, we had done all the sort of paperwork and had the drug in our laboratory to start studying it. And it turns out that their drug is about 50 to 100 times more potent than the usual sort of thing that trail ligand that sort of usually turns this receptor on. And so this was actually a drug that was already in clinical trials at the time. And so we got it in the laboratory and we put it on our cells, and lo and behold, it was actually increasing this sort of the cell death, as you can see there in the combination of the green as compared to either temozolomide or the Inhibrx 109 is what it's called alone. You can see here just increasing cell cytotoxicity or cell death as compared to either single agent. And when we do what's called synergy analyses, we're basically trying to figure out, is it additive, meaning like 1 + 1 = 2, or is it synergistic, meaning 1 + 1 might equal 4? It turned out actually the combination of the 2 drugs was synergistic together in the laboratory. And so what we kind of ultimately figured out in terms of this sort of bench to bedside is that this temozolomide was causing this DNA damage that we knew about, was causing this sort of mechanism of ER stress that I won't get into, but ultimately turning on this DR5 gene, which then when we hit the cells with Inhibrx 109 would actually increase the cell death. So what was done at the time, this was now just a couple of years ago, we expanded this as part of the phase 1 clinical trial that Inhibrx was running. This trial was open at many sites around the country, including here at MD Anderson, at OHSU, in Portland, as well as in San Diego. And I just show one example of one of the patients that was treated on the trial here at MD Anderson. It was actually a young 17-year-old male with an SDH-B mutant GIST. You can see here on this is the coronal images, but on the axial ones that here is that he's got fluid around his liver. He's got a sort of large tumor here, as well as large tumor here in his pelvis. And this was a patient treated by Dr. Samaya and Dr. Hong here at MD Anderson. And what you can see here is after just a mere 10 weeks of therapy, so 2.5 months, he had a dramatic response to therapy with this combination of temozolomide plus Inhibrx 109. Actually, so that therefore leading us to really sort of now ultimately get funded for a consortium of 10 institutions for what we've, this is funded through the National Cancer Institute, for an sdh-deficient GIST translational research and clinical trials consortium that basically joins everything from clinicians to researchers to patient advocates to industry partners to conduct clinical trials. And the first trial we're now working on, sort of moving to the next steps, is doing the phase 2 trial of the SDH, or of the temozolomide plus Inhibrx 109 in patients with SDH mutant GIST. Some of the other things that we're working on is trying to personalize therapy and trying to really better understand how to prognosticate, and also making new models of GIST so that we don't just have those 3 cell lines I just described to you, but we're developing multiple other cell lines in the process. Now, this has been an exciting time for the SDH field, because also around the same time as we were working on our work, the group in Boston was doing some additional work in their laboratory at Dana-Farber. And what they had identified is actually that the SDH deficiency and this DNA hypermethylation that I mentioned to you causes upregulation of expression of the fibroblast growth factor receptor, or ligands, excuse me. So these are basically the things that engage that receptor that I talked to you about earlier for those gene fusions. And so it causes upregulation of the FGF3 and FGF4. And what that actually led to was another clinical trial run out of the National Cancer Institute. This was a phase 2 trial of a study called regorafenib. This was in soft tissue sarcomas, but with an SDH-deficient GIST arm. And exciting to say, they actually had 10 out of 24 responders, so an objective response rate of 41 or 42%. So this really sort of now opens up another promising opportunity for a second potential drug. The toxicities were expected, and they sort of were consistent with other FGF inhibitors, and they were mild to moderate. They basically showed sort of that this mechanism, the mechanism that they're working on. And they ultimately did this actually in a very short period of time by enrolling this with multiple sites. Now, the unfortunate thing was this drug regorafenib was being made by Bayer at the time, and Bayer decided that they were not going to pursue further development of this drug. So what Dr. Suzanne George, who was leading this trial at Dana-Farber, is doing is now currently working on trying to develop a trial with a different FDA-approved drug called pemigatinib, which is another FGFR inhibitor, to study in these patients. So the third sort of exciting movement in this field over just the last essentially 2-3 years now is a third drug called olverembatinib. This is a drug that was made in China. This was a drug that was approved in China for chronic myelogenous leukemia, actually the same disease that imatinib was originally developed for. Interestingly, one of its targets, you can see here, is FGFR as well. And what you can see here in this study is that of the 26 patients they treated, they had 6 partial responses as well. There's been some other anecdotal reports as sort of we're explaining to people about the role of temozolomide. There was a group at Yale that actually treated a patient with a combination of temozolomide plus another drug called olaparib. They had this nice response to therapy in a patient. This was another study on the right side of imatinib plus binimetinib, which is a MEK inhibitor, sort of like that, similar to that drug that I mentioned that's being used for the BRAF mutant GIST. Out of 5 sdh-deficient GIST they had 1 responder. So unfortunately, it's a little bit fuzzy, at least on the screen here. But what you can start seeing here is that we have gone from just a couple of years ago, really not having great drugs available, or having essentially no drugs available that were specifically for SDH, to starting to see some disease response and some movement in the field. And so I think that we've got more work ahead of us, but we've certainly made a lot of progress just in the last 5 years in the SDH-deficient GIST field in terms of trying to identify new therapies for patients. So in the last little bit, I'm going to talk about a few other things, sort of, you heard about the role of surgery, and there's been a lot of debate actually in the SDH field, like, how do we manage these patients? Do we wedge resect them, meaning just kind of carve out like the tumor like you saw in those images? Do we do larger resections, like a partial gastrectomy, meaning like take out like a bottom part of the stomach? Do we need to take out lymph nodes in these, either selectively sort of cherry picking lymph nodes, or do we just need to clear all the lymph nodes? There is no answer to any of these questions. There is absolutely zero data. It's something that we're working on now to try to answer some of these questions, at least in a retrospective fashion. But I think the one thing we can say for these patients is they should not be getting total gastrectomies, meaning cutting out the entire stomach. That was a discussion many years ago, and I think if anything else, what I would say is from the data that we published describing the location of SDH-deficient GIST, and what you can see here is they're mostly in this lesser curve, greater curve, and in the antrum, is that you don't see SDH-deficient GIST, up in the top of the stomach very often. I'm not going to say never, because I've seen it subsequently to publishing this, but it's rare. But that said, for that one-off rare case here and there, it doesn't make sense to be thinking about doing total gastrectomies on these patients. The next question is a huge area of debate. What about metastatic disease? The group from Boston Children's and the NIH for many years sort of were proponents of only doing palliative resections, meaning if somebody was bleeding, obstructed, perforated, then they'd get an operation. Versus my sort of approach or description was, we described doing selective resection, meaning selecting the right patient based upon disease biology, extent of disease, morbidity, et cetera. And we actually did an analysis looking at adolescent and young adult patients, not specifically in SDH, because there's not a way in any of the national databases to do that. But we could see as sort of as a surrogate based on age, is that younger patients that had surgery or resection did better than those that had no resection. If we go back and look at that data of our combined data with the NIH as well as with liferaft data, what you can see here is that, as we sort of get beyond no, 1, 2, or now we're beyond 2 drugs, outcomes start getting worse as you start having more and more drugs, which is consistent with the fact that people are failing one drug, then another, then another. But what you can see here on the right side is those patients that had no operations in blue did not fare as well as those patients that had 1 or more operations, as you can see there in yellow or red, suggesting that there may be a role for managing these patients with surgical resection. When we looked at all the predictors of worsened overall survival, it was older age, being male, sort of more advanced disease, more therapies, not surprisingly, just as they were feeling them, and doing no surgery. And so one of the things that we think about as surgeons and as medical oncologists is, how does disease control how does managing with the disease control sort of affect our decision making? And you heard from Dr. Scally about that, the ideal situation is patients that have responsive tumors or they have stable disease, as opposed to those that have unifocal or multifocal progression for doing cytoreductive surgeries. And so what I show you here is actually 5 patients from the temozolomide trial. What you can see here is these are the same 5 patients that are actually on all 3 curves. The green curve is their progression-free survival, meaning how long it took, how quickly their disease was moving along before they started temozolomide. And basically, they had their progression-free survival immediately prior to starting on temozolomide was basically a median or an average, if you will, of 3 months. When we put them on temozolomide, we sort of shifted the biology of their disease, and the progression-free survival was 1.3 years. In these 5 patients, which were obviously highly selected, I then took them to the operating room, cleared all their visible disease that I could see, and we actually now shifted it out to what we consider the recurrence-free survival, was almost 2 years. So what you can see is sort of with a combination of drugs, surgery, multidisciplinary care, we can start shifting the biology and the natural history of these diseases. So what are some other local therapies that we can do? And so this is unpublished data as well that I'm going to show you. We're just finishing off the paper right now. We did present this at the Connective Tissue Oncology Society meeting earlier, or at the end of last year. But one of the other therapies that we've been looking into over the last several years is using selective internal radiation therapy, or SIRT, with yttrium-90 spheres. So these are basically the sort of the chemoembolization you heard about, mention of earlier, basically shooting little beads, but rather than beads that either just block the blood supply or beads that emit little chemotherapy, is these actually emit small amounts of radiation. And so what we looked at was actually we pulled data from our center at UC San Diego, as well as from the UK and from Germany, and actually identified 10 patients that between all of us, that we had treated with SIRT over the last many years. 70% of these were female, 30% male, meaning the mean age was about 24 years old. You can see a mix of A's, B's, and C's. 9 out of the 10 patients had had some type of prior surgery prior to this. And 7 out of 10 had had some prior systemic therapy prior to this, with often many tyrosine kinase inhibitors that you're familiar with now. How do we treat these patients? Basically, a catheter goes into the groin or sometimes up in the arm, and it goes actually up to the liver and actually injects these little radioactive beads up into the liver. In the setting of these patients, we actually did one side of the liver, so the liver has a right lobe and a left lobe, so we did unilobar, meaning one lobe in 30% of patients, and bilobar in 70% of patients. The only complication we had was one patient developed cholecystitis, or inflammation of the gallbladder, requiring a gallbladder removal, but otherwise the patients actually did really well with this procedure. And what you can see here is just in one example of this very large tumor in the liver that was then embolized, and you can see here, as you've sort of seen in lots of other images, this dramatic shift from sort of this more cystic, sort of less dense looking tumor. If we actually look at our data, we actually have had 70% of the patients actually had, by this radiology RECIST criteria that we've been talking about, actually had either partial responses or complete responses. So we've actually seen, if you look at the sort of all this data that's accumulating now, we're actually, for liver-only disease, we're actually talking like a 70% disease response rate in the liver, with all of them having some type of shrinkage. Even the ones that didn't have up to 30% had some degree of shrinkage. So we're just in the process of putting this publication together for submission, but will give us another option potentially for these patients, hopefully. And in the last couple of minutes, I'm going to talk about the sort of the central dogma and the future of where things are going. And I want to thank Ginger and Just Support International, who actually helped fund some of this work and got us really going on this project. So the central dogma of biology is really this concept of DNA becomes RNA, becomes protein. And we've done a lot of talking about mutations in this DNA. And we've talked about the proteins that are expressed, but the intermediate step in between all of this is the RNA. And so one of the things that we can do is we can actually study the RNA. But one of the new technologies that's come out that's just incredibly powerful over the last several years is our ability to actually not just look as a sort of bulk of a tumor and say, okay, the RNA expression in this, is as a group. Meaning, like, if I looked at this room, I could just say as a group, we're 150 humans in here, and sort of I see a mix of all these different colored T-shirts and shirts and pants, etc. what we can actually start doing is actually individually describing every single cell or every single person on a one-off basis. And so we actually collected 22 sdh-deficient GIST, which, as you can imagine, is not easy for a rare disease. And we started actually looking individually at the cells and looked at the RNA expression and could start doing sort of more sophisticated analyses. This is actually all the sort of the patient information, and we started looking at SDH-A, SDH-B, and SDH-C patients. We rarely see the SDH-Ds. I think, and honestly, in my practice, I've only seen one SDH-D mutant GIST in my practice ever, so they're incredibly rare. We can then do these plots where we can basically start understanding these different cell populations. We can identify tumor cells. We can identify, for instance, hepatocytes from tumors that had metastasized to the liver, to smooth muscle cells, to fibroblastic cells, to immune cells. We can then start sort of mapping these back to what the mutations are and identifying sort of these cell populations. If we actually look at what we call differential gene analysis, where we actually compared the SDH expression overall versus the KIT mutant tumors, as a very reassuring finding, is actually the biggest thing that we found as a difference between the two populations of tumors was that FGFR4, or the FGF4 that I talked to you earlier about. That was the target of that rogoratinib trial. We then used sort of artificial intelligence or machine learning to actually then start figuring out sort of different states of the cells, meaning trying to sort of group them together to try to understand more about the sort of individual biology of cells or combinations of cells in these tumors. And then what we identified is actually 4 different states using this approach. And these states are actually, we've called so far, they're just state 1, 2, 3, and 4. These states are actually have associations or whether they're direct or inverse associations with some of the common features that we think about. So for instance, these state 1s we think are going to probably tend to be more indolent tumors. They actually have an inverse relationship with the mitotic count. So the higher state 1 you are, the lower the mitotic count is, you can see here. State 4 over here, which is a little bit hard to see, has a direct correlation with mitotic count. So the high state 4 tumors, you can see like right here, the second one has a mitotic index of 200. And then finally, the state 3, we can associate with sort of how many lines of therapy these patients have had. You can sort of start seeing here state 3, 3 to 5 lines, down here 2 to 4. So perhaps suggesting that perhaps these different states might be identifying or differentiating, selecting for different populations of cells. We can then take that a step further and start trying to understand what sort of describes these different states in terms of the expression profiles, whether it be sort of metabolic sort of signatures or sort of migration invasion, meaning sort of markers of potentially metastatic-like cells, to neuronal type cells, to sort of more mitotically active type cells. And again, we're sort of finding some of these common signaling pathways, but we're also finding a lot of unique signaling pathways. So I think, what I've shown you over the last 45 minutes, hour or so is really sort of how genomics is broadening our understanding of GIST drivers, especially in the non-KIT, non-PDGFR mutant GIST. What I've described to you in the last 5 or so years is really some tremendous advances in the science of sdh-deficient GIST, really having now identified 4 novel therapies potentially for these patients, whether it be temozolomide, an FGF inhibitor, the ulverembatinib, or the Y90 selective internal radiation or SIRT therapy. But we still have work to do. These better therapies may also create opportunities for more role for surgical cytoreduction in these patients as we sort of understand more about the natural history. We're learning that we can start using the machine learning or artificial intelligence approaches to identify these different transcriptional programs in cells so that we can then start perhaps over the long term, start using this information to understand how do we define the prognosis? How do we predict the prognosis in patients so that we can a priori figure out which are the indolent ones, which are the aggressive ones, which have bad behavior, which have good behavior? How do we identify which of these patients may respond to a given drug? The temozolomide is not going to be the right drug for everybody, and an FGFR inhibitor is not going to be the right drug for everybody, but if we can start sort of figuring out who's the right person for the right drug, or the combination of drugs, like this combination of temozolomide plus the Inhibrx 109, will continue to sort of keep marching the field forward. And so we've got a lot of work to do ahead of us, but I think in the last couple of years we've made a lot of progress. And with that, there's a lot of people to thank. You sort of see some familiar faces in here. My laboratory, as well as collaborators on the original temozolomide trial, and a lot of our funding, including from GSI, as well as LifeRaft and the NIH, and a lot of people that have done a lot of work related to this. And obviously, we couldn't do this without patients and their families. And with that, I'm done. Happy to answer questions, I think, because I think we're at the last session here, right? Thank you.